Is the Zoom working, do you think? Given so and part of the key how do you throw the Zoom, can you hear me? No response?
Yes.
Yeah, okay, they can. Sounds good. Okay, back to this. Okay, keep going.
Yeah.
All good. Okay, so and really sort of the key to how that, so I was making the point that coevolution is a very creative process in evolution, and that it can lead to the emergence of all these very interesting, very complicated phenomena. And really the key to why coevolution does this is that it takes what we normally think of a static fitness landscape, and it allows for that fitness landscape to change and deform through time. And so organisms that are co-evolving are in this sort of continuously changing fitness landscape. They're exploring new genetic spaces, they're exploring new phenotypes. You know, this is obviously just a simulation in a two-dimensional landscape, but genomes are many-dimensional and they can contort in lots of different interesting ways. And so during these processes, you can get massive exploration of genetic spaces and phenotypic spaces, and the emergence of new functions or species or diversity and so forth. And so this is sort of the conceptual framework for the talk is about how coevolution deforms fitness landscapes and leads to these macroevolutionary phenomena. Before I get into the actual studies, I want to go over basically what we do in my lab. So we study experimental coevolution between phages and bacteria. The phage that we study typically, although there's different phages at the end, is lambda phage, and it affects E. coli. So these are basically the model system of all model systems. We've been studying these things for decades and decades. When we start an experiment, we start with an isogenic population of E. coli and an isogenic population of lambda, and we allow them to grow up. We just culture them in these sort of simple flask environments. So there's a limiting resource that the bacteria grows on. Phages infect the bacteria. Within a day, the bacteria begin to evolve resistance, and the phage evolves counter resistance, and they engage in this coevolutionary arms race. And what is really great about experimental evolution is that you can create this frozen fossil record where each and every day in our experiments, the dynamics are so fast that we freeze down samples every day. And you can preserve the full population of the phage and the full population of the bacteria, so you can actually reconstruct. Luckily, when you're studying microbes, you can actually resurrect them from the freezer. And so you can determine what the phenotype is, how fit they are, how the phages and the bacteria interact with each other at different time points. You can sequence their full genomes and really understand each and every step of their coevolution, reconstruct it, and figure out the processes that led to their evolution. And so in the experiment, you're just transferring 1% of the population from the flask after 24 hours to a new flask, and that process starts over, and you can see a significant amount of evolution just within weeks. The type of data that we normally collect at the end of our experiment is this interaction network data, where we have bacteria isolated from different time points, bacteria on the y-axis, and you have phage on the x-axis, where you have different genotypes of the phages isolated from different time points. And then you can create these interaction networks where you challenge each of those phage isolates with each of the bacterial isolates and construct this interaction network to see how those interactions changed over time. And what you tend to see, this is a very typical arms race, where you see the bacteria evolving more and more resistance, so more and more gray in this interaction network, and the phages evolving an expanded host range to incorporate the bacteria that they're currently infecting, and usually they're able to infect the bacteria from the past. And so this is very typical types of data that we'll collect, and you can do interesting time shift experiments to characterize how the coevolution worked and so forth. And nowadays, it's very easy to sequence genomes. It's very cheap. And so you can sequence all of those phages and all of those bacteria and get a complete reconstruction of exactly what mutations arose where, and then begin to develop hypotheses for which mutations actually influence those interactions and influence those structures in this interaction network. And catching myself saying nowadays again, I guess I'm that phase of my career where getting older, I guess, and we have new technology. Now we can do all kinds of new stuff. You can also, and this is what I learned in my postdoc, how to do synthetic biology, where you can make genetic constructs. You can have hypotheses for what mutations matter in shaping these interactions here, and then you can actually test them by taking the handful of mutations that you think matter and put them back into the phage's genome or the bacterial's genome and see if they influence their interactions. And nowadays, you don't have to just sort of select one mutation, but you can make massive genetic libraries, and you can construct what we call fitness landscapes or these networks, where you have all combinations of mutations that you think are adaptive, and you can make all combinations of those mutations and individual genotypes. You can take these libraries of phages or libraries of bacteria and study their relative fitness or other characteristics of them, so that you're not just studying sort of what is the adaptive effect of a single mutation, but actually what is its effect in combination with lots of other mutations. And this type of data, this sort of network of genotype data, is our fitness landscapes. This is what we need to be able to do to actually predict how evolution works, how adaptive evolution works, how a population goes from having 0 mutations on this side of the network to having 10 mutations on this side of the network. So we'll be looking at these networks more, but I guess maybe I should take some time right now to explain how they work. This is each of these individual nodes in the network is an individual genotype that has a combination of mutations. On the x-axis, we have number of mutations. So this is the ancestor that has no mutations, and this is an evolved type that has 10 mutations. And each of these edges or lines connect genotypes that are separated by just a single mutation. The y-axis is fitness, relative fitness. This is selection rate. And so 0 means that you have the exact same fitness ability to grow as the ancestor. Anything above that is a fitness benefit. And so in that fitness landscape, you can see that there are all these different edges and ways of sort of moving through this landscape through mutation and then natural selection as well. And you can even use this kind of information to simulate population dynamics and simulate evolution on the landscapes. So this is sort of the routine of what we do in the lab, where we evolve under interesting circumstances, the phage and the bacteria. We take all of these different steps till we really have a thorough understanding of how the evolution works. And we can test hypotheses then about the evolutionary processes. So for the first set of experiments I want to talk about, it's a study that I actually did as a PhD student that we followed up on in my lab. And we challenged Lambda to evolve to use a new receptor. The idea is that a major question in evolutionary biology is how organisms evolve new functions. And there's a lot of mystery behind this. It's thought to be very difficult to evolve. And so we wanted a model system to do that. But also in the terms of viruses, evolving to use a new receptor is an innovation in that usually is what triggers host range expansion to a novel species, and can be some of the causal mechanisms for such things as SARS-CoV-2 jumping into the human population. So we just wanted a model system to be able to study novelty, but also host range expansion in viruses. And so the work that I'm going to talk about today is largely done by ISMEJ Gupta, who is a former graduate student and postdoc in my lab. And so when I was in my PhD, we set up conditions such that we thought that there would be the right conditions to select for Lambda to use a new receptor. So basically, Lambda uses this outer membrane receptor on E. coli called LAMB. And under certain conditions, E. coli evolves to downregulate LAMB. And so we thought under those conditions, that would, the phages survive with the E. coli, but they're not doing very well because they have very few cells that actually express LAMB, very few cells to infect. And so if we waited around long enough, maybe they would evolve the necessary mutations to allow them to use a brand new receptor. And so we just set up this experiment. We had 96 separate populations. We monitored daily whether or not the phage was able to evolve to use a brand new receptor. And so sure enough, and it really changed the trajectory of my research and I guess life, this experiment worked out. And we found that in 24 out of 96 populations, Lambda evolved on average in just 12 days, so just a couple weeks, to use a new receptor. So repeated evolution, not 100% repeatable, but 1 in 4. And it always evolved to use the new receptor was OMPF. It's a structurally similar protein to LAMB, but at the sequence level, it's very different. And it evolved to use this. It took slightly different molecular routes in each of the independent populations, but there is a theme to the mutations, such that you always had to evolve 3 mutations in the sort of loop region of the protein. This is the host recognition protein, the J protein in Lambda, and also other mutations that were in the sort of more structural region of the protein that causes the protein to kind of destabilize, which we found out later was important for this innovation. And so at this stage, we were interested in sort of, what was governing the evolution of these mutations, and why was the evolution so repeatable, and how do you get, the march to doing something brand new. And so this was, when we published that paper, just our findings that we could actually see an innovation within a short amount of time in the lab. There was a commentary written by Thompson at the same time that accompanied in the same article, where we pitched this idea that the way that this evolution worked was that there was a fitness landscape that was sort of intrinsic to the phages, and that there are probably two fitness peaks, one associated with using LAMB and one associated with using OMP-F. And that when the bacteria evolves resistance, so a single resistance mutation to downregulate that LAMB, that it would open up a pathway. So now there was an adaptive pathway in order to gain the mutations to access this new receptor. So that coevolution was a sort of key feature of setting the stage for the innovation to evolve. And so this was the idea, and it seemed right, but actually this is sort of part of the caption is the figure is intended heuristically. The full fitness landscape was not measured in this study and cannot be captured in these three-dimensional spaces. So basically saying, this is a cool finding, but we don't really understand at this point how it works. And it always bugged me that we didn't really know this. And so I set out for my postdoc to figure out some technology so that we could actually measure the relevant fitness space and measure how that space deforms during coevolution, so we could see, does coevolution actually play a role driving the innovation, and is it moderated through changes in the structures of the fitness landscape? And so one of my things I love to do is to use synthetic biology to construct new technologies in order to measure things that are relevant to testing evolutionary theory. And I am happy, especially after this talk, to talk about the technologies and how to use them and so forth. But this talk, I'm just going to focus on the data and how it helps us understand how coevolution works. And so these are the fitness landscapes that we actually measured. This is the landscape. So we chose 10 mutations that we saw repeatedly evolve in that original 96 population experiment. These are all mutations in the host recognition protein, the J protein. And we constructed a genetic library of all combinations of those mutations. That technology at the time wasn't that great, and so we didn't quite get there, but we got lots of combinations. And then we were able to measure the fitness of all of these different genotypes all at once in a single experiment in the presence of the ancestral host. And so that's this sort of cartoon, or in the presence of this multi-minus, that's the resistant host that downregulates LAMB. And so from this data, what we can see just visually, we can back this up with statistics, of course, as well. But visually, we can see that the fitness landscape indeed does deform. But this is really complex data. It's 10-dimensional data. I can't really tell you, does it deform in a way that creates these sort of bridges, and does it actually promote the evolution of this innovation? And so the way that we handle that is we can actually simulate populations evolving on these landscapes and see, where do they end up? Do they end up with the right combination of mutations to use the new receptor, or do they get stuck on maybe a local peak and they don't actually end up getting to use that new receptor? In both landscapes, the 10 mute is the most fit, but it's not necessarily true that there's a pathway to get through the landscape to get to the 10 mutations, or you don't need all 10 to be OMP-F plus. You can have a subset. You could be at this part of the landscape. And so this is a representation of just one simulation. And the way that we do this is, this is a Wright-Fisher model. You have mutation, you have stochastic drift that's dependent on the population size. We use the population sizes in our experiment. And then genotypes that have higher fitness are more likely to give rise to progeny than genotypes that have lower fitness. And you can just simulate the population. The size of the red indicates how many individuals in that population have that particular genotype. And so as you can see in this one simulation, so we simulated for much longer than our actual evolution experiments. What happens is that the population gets stuck at this sort of early point in the landscape, and that's not a point yet where they have access to use the new receptor. It doesn't have the right combination of mutations. And so in our sort of cartoon picture that Thompson published, what we thought is that what we needed to do is to form the fitness landscape to create a single ridge that would take the population through the landscape. And so then when we simulated evolution on this new landscape, it really stalls out early. It does sort of propagate a little bit further through the landscape, but it doesn't tend to get all the way through the landscape to gain the mutations for the new receptor. So our hypothesis was actually wrong. And then we were sort of stuck at this point where we thought, well, what's going on? Why, how did the phages actually evolve to get to the combination mutations to use the new receptor? And so then we hypothesized that it's not about necessarily having the single right landscape, but it's about fluctuating landscapes. And the fact that in the experiment, the phages start evolving on the ancestral bacteria, and then they continue evolving on the resistant bacteria. And it's that transition from evolution one landscape that then gets you to a point that when you transition to the new landscape, you will access a new region and open up new pathways to get all the way to the end of the landscape. And so this is just one simulation or one set of simulations, but of course we did many, many more simulations. And here are sort of the overall results. And so this is frequency of OMP-F evolution. When we have just evolution on the ancestor, we never get phages that are OMP-F plus. When you have evolution with a multi-minus, you do get some simulations where you get the evolution of this key innovation. But as long as you have just a little bit of switching between one landscape to the other landscape, you really increase the probability of evolving to use this new receptor. So the way that this x-axis works is this is switching from 10% of the time on the original landscape, 90% of the time on the new landscape. And so as you see, as long as you have a little bit of jostling of that fitness landscape, you end up exploring more space and you end up getting to this key innovation much more often. And so this was our evidence that coevolution is really important, sort of changing and reformatting of the landscape to gaining this new ability. So this is all published, and there's also follow-up experiments to sort of verify this as well in the lab. But at this point, I'm going to move on to a second set of studies. And this is basically a set of studies. After you get that key innovation, you get that phage that's able to use two receptors. Then we asked, would that phage evolve now to split into specialists on one receptor, specialists on another receptor? And can we construct an environment in the lab where this would actually equate to viral speciation? And so this work is being followed up by my postdoc, Mike Dowd, in the lab. But this is also published in a paper from 2016 in Science. That's where we really first observed this phage speciation. So basically, the setup is that we take this generalist phage here, and we select it in an environment where one set of cells just express OMP-F and one set of cells just express LAM-B. And we ask, will just a good generalist evolve that can use both sets of cells, or will specialists emerge? And when specialists emerge, that indicates that there's speciation happening. So when phage co-infect the exact same cell, they recombine DNA. So there is phage sex in a sense. And so this is kind of like that Ragolitas fly system, if you've heard about. But basically, when they begin to specialize on different hosts, they're changing their ecology, but they're also creating a reproductive barrier between the different types that use the different host cells. And if there's a complete split in that you're always using one host cell or always using the other host cell, then there's no potential for genetic exchange between these two genotypes, and they're good species in terms of the biological species concept. And so we ran this experiment, and we just mixed the two cells in the exact same class. So no need for allopatry here, just sympatry. And the question is, will they develop mutations so we get diversification and specialization on these two hosts? So we ran this experiment in 6 separate replicates, and we isolated phages with one bacteria and isolated phages with the other bacteria, and asked, what is their preference for one bacterium versus the other bacterium. And we calculate what we call a specialization index. And so the way the index works is that if you have a complete 1, that means that you completely favor Lam-B, you can no longer use the cells that express OMP-F. And if you have a -1, that means that you completely favor OMP-F and you no longer use the cells that express Lam-B. And so we see again and again that in all 6 replicates, and this is the ancestral measurement in the specialization index. This just means that the ancestor is a generalist, it can use both cells, but it actually prefers Lam-B still. That's the original receptor that it can use. And so we see again and again that diversity, in fact, evolves in all of these populations. Sometimes you have what are nice, clean specialists on Lam-B and specialists on OMP-F. Sometimes you have a mixture of things that really love Lam-B and then these kind of generalists that are more like the ancestor. But we do see that 4 out of 6 populations, you get these really well-defined specialists forming. And then when we ask, what are the mutations involved in this specialization? Again, there are mutations in the host recognition protein. That makes sense. That's modifying the way that interacts with receptors on the 2 different cells. And what we can do is we can take those mutations, we hypothesize that they indeed cause specialization. So we sequence full genomes of, this is one OMP-F specialist and the full genome of another Lam-B specialist. These occurred in the exact same population, so sympatric. And we can take them and put them into a phage that hasn't evolved, just the ancestral phage. And we can see that when we create the genotype for the OMP-F specialist and the genotype for the Lam-B specialist, we do in fact see specialization for both of those receptors. So these are the causal mutations. But what is really interesting and relates to speciation theory is that when we put all of the mutations together, the OMP-F mutations and the Lam-B mutations, we create a phage that is inviable. So luckily with phages, we can actually create these genetic constructs even if they won't grow, because we can put the phage genome in the E. coli genome, make whatever bad combination we want, sequence it, see that it's there, then try to actually grow the phage. And if they don't grow, then you know that there's some kind of genetic incompatibility that's interfering with the replication. And we can do this repeated times. So we did it twice to show that every time we make this combination, there's incompatibilities. So not only are these good biological species because of the kind of pre-zygotic barrier of infecting different host cells, but they're good biological species because when they recombine with each other, they make these reproductive incompatibilities. These are Dobzhansky-Muller incompatibilities. And so after we published this paper, we had ideas about, what does this fitness landscape look like? And how does evolution play out on the fitness landscape? And in part, we were inspired by theories of fluctuating fitness landscapes again. And so basically, the way that we sort of envision speciation working or diversification working is that if you have a generalist, a jack of all trades is not very good, and that you should have selection that promotes evolution towards one fitness peak and another fitness peak, each associated with the different receptors. And this is all well and good. And if you had a scenario like this, you should get speciation, you should get diversification, because that generalist is sitting right on that sort of saddle point, that fitness valley that separates the 2 peaks. But in reality, what are the chances that your generalist genotype falls right at the sort of fitness valley, right at the saddle point? In fact, it probably follows, like I showed in the last figure, a little bit closer to being able to specialize on one or the other receptors. And so why do we expect diversification to actually happen? Wouldn't we just expect that you get evolution to specialize on the thing that it's easiest to evolve towards, and then you get stuck on that fitness peak? And so there's adaptive dynamics theory. And this is theory that says, well, okay, yes, that is the first step in the evolutionary process, but it's actually much more complicated, and diversification is much more likely than our intuition tells us. And so this is suggesting that when you get the evolution of the specialists and the population sort of shifts to really specialize on one receptor, then the fitness value of using that receptor drops down, And now the fitness landscape should contort in a way that opens up adaptive pathways to using or specializing on the other receptor, the other host cell. And so because the landscape deforms, obviously this is a cartoon, you will eventually end up with two specialists that emerge, even though they're one evolved first, you won't get stuck on that fitness peak. So this is a prediction from adaptive dynamics theory. And as far as I can tell, there's been some experimental tests, but not where we actually measure the fitness landscape and see during coevolution. Now this is coevolution not between a host and the bacteria. I should have mentioned that the hosts are maintained the same. We replenish the host, so we have that two-host environment. We don't have to worry about resistance in the host evolving. But now this is coevolution between two different competitors, and whether or not that coevolution and how competition changes through time shifts the fitness landscape in a way to promote diversification. And so we measured the fitness landscape. Now this is 9 mutations in the J protein. Some mutations evolved to use on BEF, some mutations evolved on the way to use LAM-B. And then we measured the landscape, the structure in 3 different competitive environments. This is with just the generalist ancestor. This is with an OMP-F specialist, and this is with a LAM-B specialist. And the idea is that then we can see sort of how this contorts through time and maybe changes in a way to promote diversification. You can see again, just with your eyes, and we can show the statistics, that the shape of the fitness landscapes actually depends on what competitor there is. So there's competitor by genotype interactions. And you can just sort of see that from these. Here you can see that the landscapes are very different in that there's a lot of negative genetic interactions. I said that we created 9, we have all combinations of 9 mutations, but you can see in none of the landscapes do you actually see the 9th genotype. That's because we cannot get it to grow. When we induce the cells to produce the phages just don't grow at all. So that's similar to our previous result. We know that there's lots of these really deleterious genetic incompatibilities between these mutations. And you can see all of this interesting sign epistasis. That's the sort of zigzag lines for these fitness landscapes. So when you put mutations together from one specialist to the other specialist, they have these genetic incompatibilities. And so you see a lot of genotypes that are deleterious compared to the other fitness landscape. So back to the question though. If we simulate evolution on a landscape that's just against the ancestor, do we see speciation? Or do we need to incorporate this competition and negative frequency dependence in our simulations in order to generate diversification? And so these are just 4 example simulations of dynamics of what happened in the experiment. And so on the x-axis, we have generations. On the y-axis, we have just the abundance of certain genotypes. The genotypes are characterized by a series of 0s and 1s just to indicate what mutation they have. Don't worry about that. Each color is a different genotype. And what we can see again and again in the static landscape, this is just against the ancestral phage, we do see periods where there's a little bit of diversity. This is as one genotype overtakes the other genotype, but you get the evolution of a really fit genotype that in a single genotype, not multiple different genotypes. And so you end up getting a lot of LAM-B specialists or OMP-F specialists. It depends on just the stochastic nature of evolution on these landscapes. We do have drift and the mutations are random and so forth. And so you get different outcomes, but in general, in the static landscape, you always get one genotype emerging. However, when you have a continuously changing landscape, where as the population evolves to specialize on, say, LAM-B, then you simulate fitness, you simulate evolution on a landscape that better reflects this structure rather than this structure. And this time in the simulations, we're not just arbitrarily flipping between different states, but we're having sort of gradual transitions that as the population becomes more specialized on one receptor and more specialized on the other receptor, we have a transition to one type of landscape or the other type of landscape. And so when we do this, we actually see that there's much more complicated dynamics going on, and we actually get the emergence of multiple different genotypes and nearly every single one of our runs. And these genotypes are a LAM-B specialist and an OMP-F specialist. And so just like last time, when you simulate hundreds or thousands of simulations, here are the average results. These are, again, x-axis are generations, and y-axis is now not tracking individual genotypes, but is overall Shannon entropy or diversity in these populations. Of course, in the beginning of the evolution in the static environment, you do have diversity as one genotype overtakes the other genotypes, but then you have just a single genotype persisting in these simulations. Now, if you do the same simulations, but you allow for competition and deformation of the fitness landscape, you actually get the emergence of multiple different genotypes. And that happens repeatedly in all these different simulations. So in fact, I think this is one of the first only demonstration of this adaptive dynamics phenomena where fitness landscapes are deforming in ways to promote speciation. Okay, so the third section of the talk, I want to talk about the evolution of complexity, how coevolution sort of plays a role in the evolution of complexity in ecological networks. And so this work was just actually uploaded to bioRxiv last night. It is driven by Josh Boren, who is a PhD student in my lab about to graduate, and Krista is an undergrad that works with him, and Justin Lee is a technician that works with Josh. And so this set of experiments harkens back to observations that people were making about the structure of phage bacterial interaction networks. And so this is work that was inspired by Josh Weitz and Cesar Flores, who are collaborators of mine. And so about 10 years ago, we were just characterizing what phage bacterial interaction networks look like. And we were trying to create statistics to characterize structures, non-random patterns in these networks. And we uncovered this really interesting data set where on the x-axis, we have different phages. These are different probably species of phages or very different phages from each other. The researchers were going across the Atlantic Ocean boat, picking up phages and picking up bacteria, just challenging them against one another kind of randomly to see, what do the interactions look like. And on the x-axis, we have different bacteria isolated from different regions of the Atlantic Ocean. And what is interesting is that you get this sort of multi-scale pattern. You get these modules forming. So modules defined by phages and bacteria that tend to interact with each other, but not sets outside of the module. And then what is also interesting, why it's sort of a multi-scale pattern, is that within each module, you get what is a nested structure. In this nested structure, we tend to see in phage bacterial interaction networks. This was like in the network that I showed you from the very beginning of the talk, where you have phages that have a really broad host range, and then phages that have subsets of host ranges, one within another, kind of like a Russian doll kind of motif. And so this nested pattern is common in phage bacterial interaction networks, but it's also common in mutualistic networks and in plant and animal interactions, or in sort of more traditional ecological systems. And so we're just always kind of interested in why these form. And we're really interested in, and you can get the formation of this nested pattern very easily in the lab from just a coevolutionary dynamic where phages are gaining on the host, host evolves resistance, and this back and forth. But it was a major question of, well, how do you get sort of multi-scale dynamics emerging, or multi-scale patterns emerging? And so this was sort of always a question, and we didn't know. One enticing idea is that maybe the arms race within a module proceeds in a way that you sort of get a new module forming and a new arms race happening in that module. And that you get kind of like a fractal pattern of like where these arms races are happening in sort of a local space, and then they get so far that they then blip off into sort of new modules and so forth. But we didn't have an experimental system to really understand how this worked. These modules tend to be associated with different regions of the ocean. And so it's thought, well, maybe arms races are happening sort of at the local scale to create the nestedness, but sort of at a larger scale, you get these ecological modules forming because of that sort of spatial separation. Or maybe this involves, large phylogenetic distances, long evolutionary time in order for these individual modules to actually form. And so that, I remember when they published this, and I thought, oh, that's an interesting pattern, but there's no way we could ever actually see that emerging in a lab experiment. And so for other reasons, we began to evolve E. coli with a lambda-like phage. So this is phi-21. And we were running our typical coevolution experiments, and we began to see dynamics that were really kind of compelling and very interesting. And so we started looking much more at what was going on in that system. And so what we created this sort of interaction network like we tend to do, and we can see that an arms race is taking place, up to about day 18, where the phages gain on the bacteria evolve resistance, the phages gain on those bacteria, the bacteria gain further resistance, then the phages are able to infect those bacteria, and so forth. And that happens to about day 18. This is a very stereotypical coevolutionary dynamic, post-range expansion and gaining of resistance. But then after day 18, on day 21, we see that as the phages evolve to use the day 18 bacteria, they actually begin to lose memory of phages that they were just able to infect. And so what we get is this module formation within a single flask. This is just one population, bacteria and phages evolving 10 milliliters, and we're getting these really intricate networks forming. And so we're really interested in that. We did a formal analysis to characterize the nestedness in the network and the modularity in the network. And what we find is that there are these two individual modules. This comes out as a module as well, this sort of early stage. But you can see these really well-formed modules out. These are, the day 15 and 18 phages and bacteria, and these are the day 18 and 21 phages and bacteria that tend to form into these unique modules where these interact with each other but don't interact here. These interact with each other but don't interact here. And then the question is also, is there in fact nestedness within each of these modules? And you can see by eye that there is in fact nestedness, and you can run statistics. And we repeated measurements on these different modules, and to show that it's true that they have this substructure within. And so within just 3 weeks in our experiment, we were able to show that there is this coevolutionary progression towards now these formations of modules, and then coevolutionary dynamics within the modules that created the nested structure as well. And so this paper has just been uploaded. And there's a lot of really clever genetic experiments that Josh did, but I'm not going to sort of belabor how we did these things. But please definitely check out whenever that gets approved. It's probably not in my archives yet. But just to sort of give you the picture of what's happening and how we get the formation of these different structures in the network. What happens is that here's the bacteria, and it's expressing lots of proteins on the outer membrane, but we're going to focus on 3 proteins, OMP-F, LAMB, and OMP-C. And so this is a phage. This phage is also specialized on LAMB, just like lambda is. And so it starts out by interacting with LAMB. And so this is the interaction network down here. This is sort of what's going on at the protein level. This is what's going on at the sort of ecological level. And so we have a successful interaction. But the bacteria evolve resistance. They evolve resistance by getting rid of LAMB. Different types of mutations, but all of them have the effect to get rid of LAMB. And then the bacteria evolve, or the phage evolve to actually use a new receptor. This time it's not OMP-F. The new receptor is OMP-C. And so now we have a phage that's able to use 2 different receptors, and it's in this part of the matrix. And then the bacteria says, okay, I can just get rid of OMP-C as well. And so the bacteria gets rid of OMP-C, gains resistance. And now for this phage, we have a phage that evolved to use now LAMB, OMP-C, and OMP-F. It's a phage that can use 3 receptors. It's the only case that we know of where people have evolved such a phage. So now it's a generalist, and the bacteria is in this sort of hard spot. Basically, LAMB, OMP-F, and OMP-C can all perform basically the same function for the cell. But now if it gets rid of that last receptor, has a huge fitness cost. We do see bacteria evolve that get rid of all three, but they don't take over the populations because they're paying such a penalty of not having any of these proteins on the outer membrane. So at this point, this is when the bacteria and the phage pivot in a different type of coevolutionary arms race, and that's when you get the formation of these modules. So now what's happening is the bacteria are maintaining the OMP-F receptor, but they start tweaking the receptor. They start changing the loops in the receptor. So a number of mutations evolve in this receptor, and a number of mutations evolve in the J protein, so that now, this protein can interact with these resistant OMP-Fs that have just sort of tweaked the outside of the protein. And so you do have a successful interaction. But then a new variant of OMP-F emerges that has resistance to the phages that have this form of the J protein. And then it evolves additional J mutations. And what these J mutations do is they don't just expand the host range. They create more of like a lock and key interaction, where the host recognition protein now has very specialized interactions with sets of OMP-F in this category, or sets of OMP-F variants in this other category. And so what you get is a formation of these sort of modules where they can't cross the module. I don't understand why the host recognition protein is able to evolve to use two new receptors, three completely different proteins, but then it gets held up and sort of tweaking, and interacting with the outer membrane of OMP-F. But that's certainly what happens in the experiment, something that we really need to sort of understand. But that is what the specialized interactions are, the sort of entry points into creating these modules. And then the question is, well, then what happens within a module that you get this nested structure within the module? And what Josh was able to show by making genetic reconstructions of different OMP-Fs and isolating different phages with different combinations of J mutations is that within a module, you have a little arms race that's happening just between the J protein and OMP-F protein, such that OMP-F will gain a mutation that confers resistance to this variant, but the J can counter and gains ability to use these two variants, but for some reason it can't jump to this, this very different OMP-F variant. So you're getting this sort of escalation arms race within each of these modules, but you don't get this sort of crossing back between the different modules. And so the message here is that, in relatively few days, you can get the formation of those really multi-scale, interesting, complicated ecological networks that we see when we examine phage and bacteria that are spread across the ocean. Okay, so then we have, we're up against the time because we want to have some time for some questions. I guess if I was a little bit smoother in practice, we would have gotten to this one. I'm just second author on this paper, so I'm really actually unqualified to lecture about this anyways. This is a paper by Louise Zaman, who's one of my favorite researchers. We overlapped in my PhD. He has a lab at the University of Michigan. And he actually studied digital organisms, and you can do a lot more sophisticated things with digital organisms. And he showed that coevolution, he created digital phage that infect digital bacteria. You can get coevolution such that, or you can get evolution of the digital organisms that, if they don't allow for coevolution, the digital bacteria don't evolve very complicated functions. But if you do allow for coevolution, then they evolve these genomes that have very complex traits that can facilitate complicated logical functions like equals. So if you know about Avita, then that makes sense. If not, I'm sorry. We're just going to sort of blow through this, but it's another case, and it's in this other experimental system where coevolution itself drives the evolution of not ecological complexity, or not complexity in terms of ecological networks like I just went over, but complexity within genomes and the ability to perform these different logical functions. And again, you can actually measure fitness landscapes much easier in Luis's system, and you can show that the coevolution is deforming the fitness landscape in a way that it selects for tasks with increasing complexity over time. So I guess I want to sort of just step back and say that, coevolution is this amazing force. It's a creative force in evolutionary biology. The way that it works is it creates these circumstances where fitness landscapes are fluctuating and they drive, the evolution of innovations, they drive diversification, but also the formation of biological complexity in terms of ecological networks, but also in terms of traits in at least computer programs. This is just a mural or a picture somebody painted of Darwin's entangled bank. Darwin had this intuition at the end of his book that, in fact, if you think about the sort of simple algorithm evolution by natural selection, but you layer on the potential for coevolution in these complex interactions in biological communities, you can get really interesting dynamics forming, dynamics that we can't really think of. I can only understand them in the context of measuring fitness landscapes and modeling dynamics on these landscapes and seeing sort of different outcomes. But it really is this, I think, the key to understanding a lot of how we evolve this complexity and this diversity on Earth. So with that, I want to thank my lab, of course, and we are recruiting postdocs and graduate students if anybody's interested. So cool, thank you.
Maybe you said this and I missed it, but I was wondering if you could explain why first and third experiments seem very similar in their setup, but it seemed like they had very different results. And so could you explain why the first experiments are dynamic?
Yeah, so for Zoom. So the question is, the first and third experiments were set up nearly identical to each other. The only difference in them was a little bit of the media was slightly different. That's probably not why, but the fact that we used phage 21 and not lambda phage. But we observed different dynamics. At least for the first 18 days, the dynamics were very similar. But once we sort of, after 18 days, we got the formation of specialized interactions. And the question is, why were the dynamics different? Why did we get specialization in the second experiment? And I don't have an answer to that. We think about it a lot. This lambda phage is very similar. That J protein is very similar between the two different phages. E. coli obviously had the same set of potential receptors. We have evolved lambda. We've been studying lambda for over a decade and doing these little experiments. We tried to evolve lambda with E. coli that doesn't have F. And so asking, whether or not it would co-evolve and get to a point where it begin to use, say, OMC or another receptor. And no, it just gets stuck. It's not able to expand its host range. So I'm not sure. I'm not sure why, lambda starts out with a genotype that's hard to get to OMC. I'm not sure what's unique about P21 that it could get to OMC. I assume that, I assume that there's something about how far the expansion of P21 went that then caused it to kind of be constrained to have these more specialized interactions. But I have no evidence of that or so forth. I can say that in the lambda coevolution experiment, the focus in the coevolution leaves the outer membrane and then moves internal into the cell. So there's sort of other genes that E. coli begins to mutate that interfere with replication at other stages of the infection cycle. So maybe just the fact that it sort of moves away from the outer membrane is why we don't get this sort of specialized lock and key kind of dynamics. As far as I can tell with P21, it remains on the outer membrane and playing this dance with the post-recognition protein and the receptor. So I guess that's my only answer. Question back here. Yeah, so we haven't studied the natural variation of lambda and OMP-F as much as we've focused on the phage. So we have studied natural variation in that host recognition protein J. And we see what's really cool is, so a lot of times when you do this experimental evolution, The things that mutate are not things that evolve in nature because the lab environment is so different. So for Rich Lenski's long-term lines, the genes that get knocked out or the genes that mutate in his populations are ones that do not evolve in nature. It's actually, there's sort of an inverse relationship. But in our experiments, the single positions in the post-recognition protein that tend to evolve in the phage also have higher rates of evolution in nature. They have more genetic variation than the average position in that same gene. They tend to be positions that have insertions and deletions, so really large changes. That seems to be that. What's interesting too is that in SARS-CoV-2, in the host recognition gene, the S protein, you see the same patterns of insertions and deletions in these reactive regions of the proteins. So actually, I think that what we're observing in the lab with Lambda tends to happen with Lambda in nature for sure, but also might be more general pattern to what happens with other viruses as well. So this is one time where we can make that sort of lab nature connection. Yeah. Yeah. So we've studied this in terms of LambB. So what we did, my first experiment with Lambda 12 years ago, or no, even longer now, sorry, was we altered the carbon source that we fed coli. The limiting resource was a carbon source. We gave glucose, maltose, and maltotriose. And so what LambB is used for is to transport maltodextrins, so maltose or maltotriose, across the outer membrane. And so the thought was that we would put more and more constraint on the bacteria, and that should have influenced the coevolutionary dynamics. And we actually thought that in maltotriose it would be so constrained that we'd see this sort of specialized dance that we saw with P21. But that's not what we saw. Instead, we saw the normal sort of coevolutionary arms race that we always see with maltotriose. But in the glucose experiment, well, we saw sort of one step by the bacteria, it downregulates LambB, gets rid of it completely, and sort of the game's over. And in maltotriose, you get sort of a little dance with LambB mutations, but then they just get rid of LambB as phages evolve, broader and broader usage of variants of LambB. And then the maltotriose environment, you actually get diversity, a little bit more of a coevolutionary arms race, but you don't get that sort of specialized dance that we saw between P21 and coli with a single receptor. So yeah, so you can constrain the system in different ways based on the environment and get different dynamics and get different trade-offs. So I think that relates to what might be happening in nature as well.
I think we should adjust it.
Thank you very much.